So whenever we say the biomolecules, sir, annotation can be off. Okay. Okay. So the biomolecules are prepared from the inorganic compounds. The biomolecules are prepared from which compounds? The inorganic compounds, majorly carbon, hydrogen, nitrogen, and oxygen. These are the four inorganic elements that give rise to the organic compound. Carbon and oxygen is present as carbon dioxide, and hydrogen and oxygen is present as water. The carbon dioxide and water will condense together with the help of the chlorophyll pigment during photosynthesis, resulting in the synthesis of the glucose C6H12O6, okay, in the plants. And this is the glucose, which is a monomer prepared, and this monomer will organize into a polymer called as starch, okay, which is a stored carbohydrate. Starch is what carbohydrate? Starch is the stored carbohydrate. Is it clear? So, whenever we talk about biomolecules, we have four important biomolecules to be discussed, made up of these four important inorganic elements. One of these is the carbohydrate, and all others also the derivatives of plant only. The other is the nucleic acid. So the nucleic acid includes the DNA as well as RNA and lipids. Okay, that includes fats and oils and proteins In this chapter, we basically discuss about these four important biomolecules present in a biological organism, either a producer or a consumer present in a biological organism. Okay, either a producer or a consumer. So producer is a plant through which the metabolites are produced through the photosynthesis. Consumers are all other heterotrophic organisms which are dependent upon the producers for their food. So this is a small or a brief introduction for the chapter biomolecule. Any doubts? Any doubts?
No, sir.
Okay. So since it is recorded, I can clear the slide so that whenever you get the recorded video, you can see as such. So, sir, can I clear the slide?
Yes, sir. Shall we move on to the next slide, sir?
No. Explanation is required by writing. I will
More
clear this.
Shall we go on to whiteboard, sir?
Yeah, that is fine.
Better, is it not, sir? Yeah. Yes, sir. You can clear it now, sir.
Okay. Now, we have an experiment. For the analysis of biomolecules. Okay, we have an experiment for the analysis of biomolecules. So we take a pestle and we also take a mortar. We take a tissue. If it is an animal tissue, then it is a liver. If it is a plant tissue, then it is a cauliflower. Then to this, we will add the solvent. The solvent what we add is the trichloroacetic acid. The solvent what we add is the trichloroacetic acid. Then we grind the tissue. Then what we do? We grind the tissue. We get the grinded tissue. And this grinded tissue will be passed through the test tube, Through the cheesecloth. Through what cloth? Cheesecloth. Through the cheesecloth, it will be filtered. And if you filter through the cheesecloth, all the debris will be trapped in the cheesecloth, and you get the filtrate of it. And this filtrate, sir, can you move the slide?
Yes, sir. Shall we move on to the slide now?
No. Move the whiteboard. Sir.
Okay, no. Actually, you just have to clear the screen, sir. That is the only possibility.
Okay, it will be recorded, right?
It is recorded, sir. It's being recorded.
Okay. Then I will erase. So now we have the filtrate in the test tube, and this filtrate is subject to a technique called as centrifugation, which works on the centrifugal force. You get two fractions. One is the pellet, another is the supernatant. So you get the two fractions. One is the pellet. What the another fraction is called? Supernatant. Pellet is called as acid insoluble fraction. Pellet is known as what fraction? Acid insoluble fraction. And supernatant is also called as the retinate. Okay, supernatant is also called as what? The retinate, or otherwise it's also called as what? The acid soluble fraction. So you get two fractions. One is the pellet, the acid insoluble fraction. Another is the supernatant or the retinate, which is acid soluble fraction. And what do you find in the pellet? In the pellet, you find macromolecules with greater molecular weight ranging greater than 10,000 daltons. Ranging greater than how many daltons? 10,000 daltons. And in the supernatant, you find the micromolecules. Okay, in the supernatant, you find the micromolecules. In the supernatant, you find the micromolecules, and the micromolecules will have less than 1,000 daltons, approximately around 800 daltons. So which are the biomolecules you find in macromolecule fraction, that is pellet? You find carbohydrate, you find nucleic acid, and you also find proteins. But there is an exception here. The exception is the lipids. As less than 1,000 Dalton, but it is found in pellet, not in supernatant. Why the lipids, even though if they have the molecular weight less than 1,000 Dalton, they are found in the pellet but not in the lipids? Because in reality, if it is less than 1,000 Dalton, okay, then it should be present in the micromolecular fraction in the supernatant but not in the pellet. So for this, we have an explanation why the lipids which have low molecular weight classified under micromolecules but found in the macromolecular fraction. So then what else is found in the supernatant less than 8,000 Daltons? You find sulfates, SO4 minus, phosphates, PO4 minus, and other organic compounds generally you find in the cytoplasm will be found in the supernatant fraction, but the higher molecular weight compounds will be found in the pellet fraction. Hope it is clear for you. Any doubts? Okay, ma, any doubts? Respond, please. Yes or no? I did not get the reply. Okay. So the exception is the lipids. They have molecular weight less than 1000 daltons, but found in macromolecular fraction. But found in what fraction? Macromolecular fraction. It is because the lipids is present as phospholipid bilayer, and these phospholipids, which is present as bilayer, as it exposed to water, they are non-polar. What they are? Non-polar. So they result in a spherical vesicle. They result in what? Spherical vesicle. And this spherical vesicle, it is derived by the breaking of this spherical vesicle. It is derived during the breaking of cell membrane of the cell, including the mitochondrial membrane. All this I taught you in Cell the Unit of Life. Chloroplast membrane and including nuclear membrane. All membranes are made up of what? Phospholipid bilayer. This phospholipid bilayer, when they are broken, okay, in an intact cell and exposed to water, they result in spherical structures because phospholipid, as we discussed in the Cell the Unit of Life, only the head is polar, but the tail is what? Non-polar. Only the head is polar, but the tail is non-polar. So only the head region of the phospholipid will interact with the solvent, and the tail will be embedded inside. The tail will be embedded inside. When the tail is embedded inside and only the head is exposed to the inside and outside, then it results in a spherical structure called as micelle. Then it results in a spherical structure called as what? Micelle. These micelles are so tiny that they get trapped. The micelles are so tiny that they get trapped within the macromolecules. They get trapped within what molecule? The macromolecule. Therefore, all these spherical globules of lipids, they get trapped within the macromolecule, and therefore they are found in the pellet fraction, even though their molecular weight is less, okay, of less than 1000 daltons. Hope it is clear for you. Why its molecular weight is less? Even then it is found in the pellet fraction, but not in supernatant. Done. Any doubts? Any doubts?
No, sir.
Understood? Okay. Sir, we can go back to slide.
Sure, sir.
Elements such as calcium, magnesium, sulfate, phosphate, and all this calcium, magnesium, sulfate, phosphate, what did I tell you? It is found in the acid soluble fraction with molecular weight less than how many daltons? 1000 daltons. And these are also called as micromolecules. In specific, we can say biomicromolecules, what we can call it as biomicromolecules. And if you do the analysis of the ash, you also find elements such as carbon, hydrogen, oxygen, fluoride. Whereas analysis for organic compounds, analysis for compounds give the organic and inorganic constituent present in the ash. Next. See, what did I tell you which is found in abundance in human body? Hydrogen, oxygen, carbon, nitrogen, right? See, this is the percentage weight that is present in the earth crust and this percentage weight present in the human body. Amount of hydrogen present in the earth crust is 0.14, but in human body it is more, 0.5. Carbon it is 0.03, but in human body it is high, 18.5. Oxygen it is 46.56 in earth crust. In human body it is 65. Nitrogen it is very little because it is a gaseous element. Nitrogen is what element? A gaseous element. Therefore, it is found in atmosphere, not in earth crust. Therefore, very little. But in human body it is 3.3. Sulfur, they are also the component of amino acids. And where do you find the amino acids? Proteins. So since the sulfur is a component of a protein and some vitamins, it's found less in earth crust, but more in human body. All these 5 elements, hydrogen, oxygen, carbon, nitrogen and sulfur, they are found in abundance. Very, very important question for NEET. Okay, they are found in abundance in what? In the human body. Okay, but look at the other elements. Sodium, more in earth crust, less in human. Calcium, 3.6 in earth crust, less in human body, 1.5. Magnesium, 2.1 in the earth crust, less in human body, 0.1. Silicon, 27.7 in the earth crust, less in human, negligible. All this data has been derived from the scientist C.N.R. Rao and his analysis. This is a comparison of earth crust and human body. Very, very important for the NEET examination. Several times the question has been appeared from this table. Hope you have understood. Next. See, similarly, we also have a table representing the component and its formula. For sodium, we represent it as Na plus. All this is very simple for you, but still required to tell. I am telling you. Potassium K plus, calcium Ca plus plus, magnesium Mg plus plus, divalent ions. Water H2O, compound sodium chloride, calcium carbonate, phosphate and sulfate. PO4 3 minus and SO4 2 minus. Next. Okay. So before we shall go to the amino acids, let me ask you some questions. Elvin, tell me, which is a solvent that we use to grind the liver tissue or cauliflower?
Trichloroacetic acid.
How many fractions do we get?
2 fractions.
Which are those?
Pellet and supernatant.
So that is sufficient. What is another name of pellet?
Acid soluble.
No, 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 no. Pellet is the other.
Okay, acid insoluble.
Acid insoluble. Also, the pellet is also called as retentate. I hope I did a
Yes, sir.
mistake. I hope I did a mistake when I was writing. I wrote retentate for supernatant, not for pellet. Okay, please rectify that mistake. So retentate or pellet or what? Acid insoluble fraction. And what is other fraction, Elvin?
Sir, acid soluble fraction.
Or also called?
Supernatant.
Okay, that's it. Only 2 names. Which compounds you find in the supernatant?
Supernatant we find micromolecules.
Okay, less than how many Daltons?
1000.
Okay, good. Who are the others attending this meeting?
Sir, we have Evelyn, we have Levin, Ranika, Ritvik, Darwin, and Jekolia.
Sir, randomly you call anyone name, sir, to ask the questions.
Okay, ma. Levin, come on, Levin, unmute yourself.
Yes, sir.
Yes, Levin. What is the molecular weight of pellet fraction?
So molecular weight,
Yeah, I wrote a value.
so less than 10,000 Dalton.
More than
Oh, sorry. More than 10,000.
more than 10,000 daltons is macromolecular fraction. What all you find in pellet fraction? Which biomolecules you find?
So carbohydrates, nucleic acid, then proteins, lipids.
Lipids should be in supernatant or pellet.
So pellet, even though it has less than 1,000 Daltons.
Why?
So I'm not sure about that.
Why in the sense when the cell is disrupted, right? Look here. If this is your cell, listen here. All of you listen. This is your nuclear membrane. Do you agree with me? And this is your what membrane? Students? Cell membrane. All membranes are made up of what? Phospholipids only. All membranes are made up of what? Phospholipid. You disrupt the cell and break into pieces. Do you agree with me? And when you disrupt the cell and break the membrane into fragments, this phospholipid is non-polar. It is not miscible in water, and it can dissolve only in the solvents. And it makes sure that whenever the membrane is disrupted, the disrupted membranes will result in small vesicles. Each disrupted membrane will result in what vesicles? Small vesicles. Every small vesicle is called as what? Micelle. What it is called as? Micelle. It is so tiny vesicle. It is so tiny vesicle that it gets trapped. Where? Within macromolecules during centrifugation. Okay, it gets trapped within the macromolecules during the centrifugation. That is the reason why the lipids are found in what fraction? Pellet fraction. Is it clear now?
Yes, sir.
Understood. Now can you tell me when the cell membrane or nuclear membrane made up of what lipid?
Phospholipids.
Okay, when it break into fragments, it results in tiny vesicles called as
Micelle vesicles.
micelle. They are the vesicles, globular structures. They get trapped within what?
So they get trapped between the macromolecules.
And hence they are found in
That's why they're found in the pellet fraction.
pellet. Yeah, even though their molecular weight is less than 1,000 daltons. Understood? Hope it is clear for you.
Yes, sir.
Okay, good. Okay, you can unmute. Next, sir, anyone student for this lecture?
Yes, Ranika.
Yes, sir.
Okay, ma. So, which weight do you consider for analysis of elemental composition, inorganic elements? Dry weight or fresh weight? Which one you consider?
Dry weight, sir.
Why dry weight? Since the dry weight do not have what content? The moisture content. Yes or no?
Yes, sir.
Since the dry weight does not have the moisture content, you always consider the dry weight. And out of all the elements, whatever we discussed, which are those 5 elements which are more in human body than others?
Hydrogen, carbon, oxygen, and
Nitrogen
nitrogen, sulfur, sir.
and sulfur. Okay.
Yes, sir.
Okay, ma. Okay. You can unmute. You can mute. Sir, whiteboard.
Sure, sir. One second. Yes, sir, you can write here, sir. No problem.
Okay. See, the next it is amino acids. So whenever we talk about an amino acid, as you know very well, the carbon has 4 valency, and to one of the valency, there will be attachment of NH2. NH2 is the amine group of amino acid. Other is COOH. COOH is the carboxylic acid of the amino acid. Okay, COOH is the carboxylic acid of the amino acid. And the other is H, hydrogen. The next will be the R group. The R group can be an aliphatic group or it can be aromatic group. What is aliphatic? Open chain compounds. What is aliphatic, students? Open chain compound. And what is aromatic? Aromatic is the closed chain compounds. So what is open chain? Methyl, CH3, ethyl, C2H5. Closed chain carbons are such as benzene, toluene, and others. So you find all such functional groups are open chain structures or closed chain structures. And if all the 4 valence is satisfied with the carboxylic acid amine group, hydrogen, and an R group, you have 20 different amino acids. Okay, you have 20 different amino acids based on the difference in the R group. Based on the difference in the R group, you have 20 different amino acids. In textbook of NCERT, they have given 3 examples for you that these 3 functional groups will remain the same. COOH will remain the same, H will remain the same, NH2 will remain the same. What change from one amino acid and another amino acid is R group. If R is hydrogen, then it is called as glycine amino acid. If R is methyl, CH3, then the amino acid is called as the alanine. If R is hydroxymethyl, CH2OH, what do you call this CH2OH as? Hydroxymethyl, then you call such amino acid as serine. You have just represented 3 R groups as mentioned in NCERT textbook. If you type in Google images, you find all 20 different amino acids, where in all the 20 different amino acids, these 3 functional groups will remain the same. But what is different from one amino acid and another amino acid is just the R group that makes 20 different amino acids that contribute for the structure of the protein. Hope it is clear for you. Next. So accordingly, we have classified amino acids into different groups, okay, based on its nature. So what are those amino acids based on its nature? We call acidic amino acid. All these are very important. Okay, we call it as acidic amino acid, right? If it is acidic amino acid, then the example is glutamic acid. All these are very important for NEET. Then it is a basic amino acid. Then it is lysine. Okay. Then if it is a neutral amino acid, what do we call, students? Valine. Okay. And this COOH and NH2 do exhibit variation at different pH. Okay. It may change into carboxylic acid, can change into carboxyl, and amine group can change into ammonia. Okay, or even ammonium also, NH4 plus. So based on this, we have 20 different amino acids. Some of this are mentioned as acidic, basic, and neutral. Hope it is clear for you. Next.
Excuse me, sir.
Yes.
So, what are the acidic and neutral amino acids, sir?
I'll tell you regarding this. I'll continue.
No, sir. Can you just tell the names, sir?
You want a list of all 20 different amino acids and which is classified into acidic,
No, no,
basic,
no,
and
no,
neutral.
no, sir. Can you just spell the acidic and neutral amino acid?
Spelling you want.
Yes, sir.
Okay. I'll write again. GLUTAMIC ACID, glutamic acid. Neutral amino acid is VALINE, valine.
Yes, sir.
Okay. This acidic amino acids can migrate within a pH range of 1 to 6. The basic amino acid will migrate within a pH range of 8 to 14, and neutral amino acid will stop migrating at a pH of what? 7. Based on its pH, it will migrate in the gel, and we have a specific gel to separate the proteins based on its pH. Those things you study in higher classes, and we get respective bands. Okay, we call it as polyacrylamide gel, students. What do we call? Polyacrylamide gel. What do we call it as? Polyacrylamide gel. We have two electrodes, cathode, anode, and we have the well. And to separate the proteins, we'll have a suitable buffer containing ions in it. When we apply electricity and load the proteins, some proteins there will be a standard reference 1, 2, 3, 4. Finally, it will end with what? 14. But let me take it as 7 here. Those proteins which become static at 7, pH 7, and do not migrate any further, they are the example of what amino acid? Neutral amino acids. Okay. Why they do not migrate beyond pH 7? Because they are neutral in nature, and therefore they result in the formation of the band at pH 7. If they are acidic amino acids, based on the number of carboxylic acid, they may result in the band of 3, 4, 5, 6. If it is a basic amino acid, based on the number of amine groups, they can result in the band from 8 to 14. Hope it is clear for you, right? So based on the carboxylic acid group, it will attain the acidic nature. Based on the amine group, it will retain the basic nature. If both carboxylic acid and amine group, if they neutralize each other, okay, it results in the band at pH 7 and does not migrate further, they become the neutral amino acids. Is it clear? Shall I erase? Shall I erase? Okay. Next. See, we call it as zwitterion. What do we call it as? Zwitterion. See the nature of this zwitterion: carbon, hydrogen, R group. See, NH3 plus and COO minus. The carboxyl and the ammonium group, they neutralize each other. They neutralize each other. Okay. When they neutralize each other, they result in a band at pH 7, and such pH is also called as the isoelectric pH, where the proteins do not migrate any further, which contain such zwitterions or the neutral amino acids, okay, which neutralize each other by the contribution of proton from carboxylic acid to the amine to become ammonium and carboxylic acid to become carboxyl. Okay, COO minus is carboxyl and NH3 plus is ammonium. Okay, so at a physiological pH, it can be of different forms. One of the form is Ritter form. This is B. Let me see the A form. In A form, it is C, H, R, COOH, NH3 plus. Then it will be basic. Then C, H, R, NH2, COO minus. Then it will be acidic. So when base and an acidic functional group, when they neutralize each other, then it will be neutral. Such ions, neutral ions, are called as bitter ions, and they form a band at pH 7, and pH 7 in which it results a band is called as an isoelectric pH. Hope it is clear for you. Next. Sir, we shall move on to the slide.
Sure, sir.
Okay. See, as I explained you, structure of an amino acid, there are 4 substituent groups occupying 4 valence carbon position: hydrogen, carboxyl, amino, and the variable group. Variable group is the R group, which is different from each other and are responsible for 20 different amino acids. Based on the nature of R group, there are several amino acids. How many? 20. The R group can be hydrogen. If it is hydrogen, then it is glycine. If it is methyl, then it is alanine. If it is hydroxymethyl, then it is serine. Yes, sir. Next slide. See, everything is same. In all amino acids, 20 different amino acids, these 3 functional groups will be the same. This will be different. If R is hydrogen, glycine. R is methyl alanine. R is hydroxymethyl serine. Next. So as I told you, based on the number of amino group or the carboxylic acid group, NH2 and COOH, you can have acidic amino acid glutamic acid, basic amino acid lysine, neutral amino acid valine. Aromatic is those which contain the closed ring structures. It can be tyrosine, phenylalanine, and tryptophan. Next. Yeah, look here. So particular property is the ionizable nature of NH2 to become NH3 plus and carboxylic acid to become COO minus. In solutions at different pH, the structure of amino acid changes. It will not remain the same. Therefore, Zwitter ions are those which have both positive charge and negative charge, which neutralize each other. Okay, and this is your Zwitter ion B. B is your what ion? Zwitter ion with positive charge on the ammonium group, negative charge on the carboxylic acid group. Where in A, you have only positive charge on the amino group. In the C, you have only negative charge on the carboxyl. Next. Okay. Sir, empty slide, empty slide. Okay, we'll explain the lipids, then I'll ask the questions together. So lipids are generally, just now I was talking about lipids. Lipids are generally water insoluble. or we can say it as what? Non-polar, students. Lipids are water insoluble, or otherwise what we can call it as non-polar. So this non-polarity of lipids is because of the phospholipid. The phospholipid found in cell membrane. This was I am talking about earlier. Now it will become much more clear for you. Is called as lecithin. What do you call it as? Lecithin. 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 There is a glycerol. CH2OH. CHOH. CH2OH. This structure with 3 carbon atoms, carbon 1, carbon 2, carbon 3. You call it as glycerol, or you also call it as trihydroxypropane. So what do you also call it as? Trihydroxypropane. To the glycerol, you will add fatty acid. To the glycerol, what you will add? Fatty acid. The general formula of the fatty acid is RCOOH. What it is? RCOOH. Now let us see what happens to the glycerol when a fatty acid is added to it. So the output is, if the glycerol is attached to fatty acid 1 to carbon 1, fatty acid 2 to carbon 2, and fatty acid 3 to carbon 3, okay, then it is called as a triglyceride. What it is called as? Triglyceride in general. If it is 2 fatty acids, diglyceride, and if it is 1 fatty acid, monoglyceride. So you have 3 different types: triglycerides, monoglycerides, and what? Diglycerides, based on the number of fatty acids attached. If glycerol is attached to 1 fatty acid, okay, if glycerol is attached to 1 fatty acid, CH2O fatty acid 1, CHOH, CH2OH, then it is monoglyceride. What it is? Monoglyceride. If glycerol is attached to 2 fatty acids, fatty acid 1, fatty acid 2, then it is called as diglyceride. If glycerol is attached to 3 fatty acids, fatty acid 1, fatty acid 2, fatty acid 3, then this structure is called as the triglyceride. So whatever the food we take, generally we take it as triglycerides. Get digested to diglyceride. Diglyceride get digested to monoglyceride. Finally, monoglycerides get digested to glycerol and fatty acids. But in phospholipid, the 2 carbon atoms are attached to fatty acid 1 and fatty acid 2, but the third carbon atom is attached to a phosphate group. Therefore, it is called as phospholipid, along with other functional group called as, okay, lecithin. Other functional group where there is an attachment of CH2, CH2, and N, CH3, CH3, CH3. When you have such functional group, such phospholipids are called as what? Lecithin. Such phospholipids are called as what? Lecithin. Okay. And these phospholipids, you find it in cell membrane of mitochondria, nucleus, including cell envelope, plasma membrane. Now, of all these 3 carbons, only 1 carbon is polar. That is third carbon containing phosphate group. The phosphate group will be polar. Therefore, the PO4 minus will form the head. Whereas the two fatty acids of carbon 1 and carbon 2, they become the tail. That is the reason I told you. Fatty acids that result in tail attached to carbon 1 and carbon 2, they are non-polar. Whereas the head is made up of the phosphate group attached to the third carbon, which is polar. So this is the general structure of phospholipid with head and how many tails? Two tails. Only head is polar, tails are non-polar. Hope it is clear for. Next.
Excuse me, sir.
Yes.
Sir, can you explain this lecithin again?
See, lecithin, there are different types of phospholipids. Okay. In neuron, we call it sphingomyelin. In cell membrane, we call it as lecithin. All in general contain phosphate group attached to third carbon. Is it clear? Fatty acid 1 and fatty acid 2 attached to carbon 1 and carbon 2. Clear? What makes difference between one phospholipid and another phospholipid is the additional functional group, which has been shown in what? The bracket. N having 3 methyl groups. If you see the other phospholipids, the functional group will not be the same, it will be different. In sphingomyelin, it will be different. In cerebroside, it will be different. Understood? Other things will be the same. Fatty acid attached to carbon 1 will be the same. Fatty acid 2 attached to carbon 2 will be the same. Phosphate group will be the same. The additional functional group, what you find here, will be different. Will be different from one phospholipid to what? Another phospholipid. Is it clear?
Yes, sir.
Here, this functional group, which is attached to the third carbon, is called as what? Lecithin. Or it is called as lecithin. Understood?
Yes,
Here,
sir.
if you want to show this phospholipid diagrammatically, the phosphate group attached to the third carbon is the head. The fatty acids attached to carbon 1 and carbon 2 are the tails. Since phosphate group has a negative charge, head is polar. Fatty acids do not have any charge. Therefore, tails are what? Non-polar. Understood? Is it clear?
Yes, sir.
Okay. Fine. Next. Now, if I talk just about fatty acid, if I just talk about fatty acids, okay, the fatty acid has a general formula R COOH. The R can vary from methyl, CH3, ethyl, C2H5, propyl, okay, butyl, pentyl, hexyl, whatever you have studied in chemistry. The R can range from carbon 1 up to carbon 19. So in diagram of the textbook, they have given one example, students, called as palmitic acid. In the textbook diagram, they have given an example of palmitic acid. The palmitic acid is a 16 carbon saturated fatty acid. What do you mean by saturated fatty acid? Only with a single bond. What is saturated fatty acid, students? Only with what bond? Single bond. So how do you diagrammatically represent CH3, CH2, 14, COOH. So 14 plus 1, 15, 15 plus 1, 16. So you find many such fatty acids from carbon 1 to carbon 16. One such example that has been prescribed in your textbook is the 16 carbon fatty acid called as palmitic acid. Even here, only COOH is polar. The hydrocarbon containing carbon and hydrogen is non-polar, even including in fatty acid. So how do you diagrammatically show a fatty acid? You show with the head and one tail. Head is made up of carboxylic acid. Okay, head is made up of carboxylic acid, whereas tail is made up of what group? R group. Head is polar, tail is non-polar. Understood? Is it clear? Next, finally, we shall conclude with lipids by describing lipids are broadly divided into two types. Lipids are broadly divided into two types. We call fats and oils. What do we say? Fats and oils. Fats are saturated lipids with single bond only. Oils are unsaturated lipids with double or what bond? Triple bond. Generally, you find unsaturated lipids in vegetable oils. Okay. Sir, can you remove that? Okay, let it be. Okay.
Which one should be
Generally,
removed, sir?
the text.
Oh, text should be removed, sir. That is.
Yeah, enough. So generally, you find the unsaturated lipids with double and triple bond in vegetable oils, and therefore they have low melting point. They have what point? Low melting point, and they remain liquid. Okay, they remain liquid at what temperature? Room temperature. They remain liquid at room temperature, whereas saturated lipids, which is made up of only single bond, is found in animal fat. Animal fats, and they have high melting point. They have high melting point, and they remain solid at room temperature. So that is the reason why vegetable lipids are good for health than what? Animal source. Animal source, it get deposited in the body adipose tissue, resulting in obese condition. Okay, fine. This completes the lipids. Hope it is clear for. I'll erase it. So now you can go on to the slides. Lipids are water insoluble and could be a simple fatty acid. A fatty acid has a carboxyl group and a R group, that is R COOH. R group could be methyl, ethyl, or whatsoever from carbon number 1 to 19. Palmitic acid is a 16 carbon fatty acid which is saturated with only single bond. They are given another example for fatty acid which is arachidonic acid made up of 20 carbons. Next slide. Next slide. Sir? Sir?
Yes, sir. Next slide, sir.
Yes, sir. Yes.
Sure, sir.
So types of fatty acids: saturated without double bonds, unsaturated with one or more double bonds. Next. See here. This is all what I told you. Saturated, no double bonds. Unsaturated, one or more double bonds. Carbon atoms are saturated with single bond. Unsaturated, they have double or triple bond. Animal fats you find in saturated fatty acids. Plant fats you find in unsaturated fatty acids. Animal fats are saturated fatty acids solid at room temperature, here it is liquid at room temperature. Essential fatty acids are not present in animal fats, but essential fatty acids are present in plant fats. What is essential, which has to be taken only through diet or food. Since they remain solid at room temperature, they have high melting point. Since they remain liquid at room temperature, they have low melting point. Example of animal fats are stearic acid and lauric acid, or plant fats are linoleic acid and oleic acid. Hope it is clear for next. Okay, this is what I was trying to tell you. See, this is a glycerol with carbon 1, carbon 2, carbon 3. This is a fatty acid with R group and what? COOH. Now, how many fatty acids are added there? 3. Fatty acid 1, fatty acid 2, fatty acid 3 for glycerol, carbon 1, carbon 2, carbon 3. Therefore, it is called as what glyceride? Triglyceride. Next. Next. Okay. See here. Glycerol is also called as trihydroxypropane, I told you. In many lipids, glycerol will be in attachment with fatty acids to become a monoglyceride, diglyceride, or triglyceride. Already I told you what is mono, di, or tri. If a glycerol has to attach to a fatty acid, the bond that is responsible for the attachment of fatty acid, okay, the bond that is responsible for the attachment of fatty acid, the bond is called as ester bond. Very, very important for NEET examination. And reaction is called as esterification. And what the reaction is called as esterification. Make a note of it, very important. The fatty acids are esterified with glycerol. Next. Next. Okay. Okay, based on the melting point, sir, previously. Oh, next slide, next slide. Okay. See here. Based on the melting point, we classified it into fats and oils. Fats are solid in winters and have high melting point. Oils are liquid in winter and have low melting point. Lipids are associated with phosphorus. I told you, right? Phosphate, then they become the phospholipid. One of the phospholipids with containing the additional functional group for nitrogen with 3 methyl groups. Okay, is called as lecithin. Next. So compound lipids are also found in other tissues. As I told you, neural tissue, it is called as sphingomyelin. Next. Okay, sir. You can call anyone student to ask a question.
Yes, sir. Yes, ma'am, Evelyn? Yes, sir, you can ask the question.
Yes, sir.